So it's a real privilege to be here today and to be part of this symposium. And I have to say that there are very few neuroscientists of my age whose work wasn't influenced by the seminal work of Professor Bert Sakmann and Erwin Neher. I am definitely not those. When I started my studies in Oxford as a graduate student, every week or every month, I was reading papers coming from Bert Sakmann's lab. For example, Arthur Connors and Bert Sakmann, they wrote the paper which I first read about quantal synaptic neurotransmission in the hippocampus of inhibitory synapses. Then there were lots of papers authored by David Cohun, Bert Sakmann, Peter Jonas on glutamatergic synaptic transmission and glutamate receptors. A few years later, he started a really nice collaboration with Peter Seeburg, where they described lots of glutamate receptor function, structure, and subunit composition effect. Later on, with the work of Greg Stewart, with Mike Heuser and Nelson Spruston, those papers really influenced my development. So I really cannot tell you that how influential his work was on my scientific career. Today I'm going to show you some data which actually started on basic findings, which was published again by Bert Sakmann. And I'm going to show you those but the main topic of my talk is actually synaptic diversity, and I'm again in the privileged position that my lecture was introduced by Professor Erwin Neher talking about synaptic diversity. So the main interest of our laboratory is really understanding the molecular mechanisms underlying diverse synaptic properties in general in the brain, but we are focusing mainly in the hippocampal synapses. And this is a light microscopic sort of steady super-resolution image of a small piece of a dendrite of a CA1 pyramidal cell. And you can see that these spines, these protrusions, are of different size and shape and so on. And basically, this is the structural diversity which has been described, I think, over a century ago. And you can have even a higher resolution image of that structure by using 3D EM reconstruction. And you can see that these spines are really large and small ones, and with long neck and with short neck and so on. But that doesn't actually reflect the function of the synapse, or we don't know really what's the relationship between the structure and the function. So when I consider synaptic diversity, I have to say that I can put them conceptually in a few categories. Synaptic diversity exists between when an A cell makes a synapse onto B cell and a C cell onto D cell. So for example, the inferior olive Purkinje cell synapses, the famous climbing fiber synapse, function totally differently than the dentate gyrus granule cell in the hippocampus to the CA3 pyramidal cells. And the hypothesis is that because there are different pre- and post-synaptic molecular isoforms, and they are responsible for the diversity. And indeed, if you look at how many different glutamate receptors and presynaptic calcium channels are, or calcium sensors are extremely diverse molecularly, and all of the active zone and the release-associated proteins are diverse. I would like to point out, for example, among 13 molecules, there are 4 isoforms, and the molecule Erwin Neher was talking about. Another concept is when A cell makes synapses onto B and the C cell types, and the function is very different. Okay, so these are the famous post-synaptic target-dependent differences in synaptic properties. Today I'm going to talk about CA1 pyramidal cell to parvalbumin-positive basket cell synapses, and CA1 pyramidal cell to somatostatin-positive OLM cell synapses. And our hypothesis is that there are different molecules responsible for that, but there are also variable densities and nanoscale arrangements of the same molecules which underlie the function. And finally, it could be that A cell makes a synapse onto B1 cell, B2, and so on. And this is exactly what Erwin Neher was talking about, that one calyx and another calyx and the third calyx, they show different functional properties. And indeed, we just published a paper last year where we looked at CA1 pyramidal cell onto these basket cells, and another one and the third one, and then actually we found remarkable diversity among those synapses as well. So today, I'm going to talk about only this one. And it started with a seminal paper about 25 years ago in 1998 1978 from Bert Sakmann's lab with Alex Reyes. They patched in the neocortex of rat pyramidal cells and double recording between this, what they call this a bitufted cell. Now we know that they are somatostatin positive cells. And then they evoked action potentials in the presynaptic cells, reaction potentials. You can see nicely that the postsynaptic response called EPSP shows short-term facilitation. So the first response is small, the second is larger, and the third is even larger. And when exactly the same axon made a synapse on this multipolar cell, which we now know it's a parvalbumin expressing basket cell, then the response, the first one is large, and then it gets smaller and smaller. And this is the paired pulse ratio. Again, I remind you that Erwin Neher showed you that the paired pulse ratio for this connection is always larger than 1, and for this connection is always smaller than 1. Okay, so that was the initial observation made by Bert Sakmann's group 25 years ago, and many other groups later on, and we also described that in the hippocampus, the same synaptic connections are also functionally diverse. There are paired recordings between pyramidal cells and OLM cells here, and about 50-60 pairs, and the average responses are shown here. And when the postsynaptic cell, what I call is a fast spiking interneuron, this is the same parvalbumin expressing basket cell mainly, then there is a strong depression. And the important point is that the same presynaptic cell makes these two synapses onto the two different postsynaptic cells. And if you superimpose these average traces, you see that large, very large difference. So one of them, the amplitude on average is only 10 picoamp, and the other one is about 150 picoamp. There is a 15-fold difference in the mean postsynaptic response between these connections. And that just shows you the paired pulse ratio. The mean paired pulse ratio of these OLM cells is 2.2, and the mean paired pulse ratio is about 0.9. But I would like to point out, here you can also see the diversity of the synaptic strengths. When we look at only basket cells, some of them have 600 picoamp responses. The mean is about 150 picoamp, but there are actually very small ones. So that was the subject of our previous paper, and I don't have time to talk about what is the underlying mechanism. But when you scale these responses to this, and then you look at the postsynaptic response kinetics, they are remarkably similar. But that's not very important right now. So what could be the reason for this large difference? One of the reasons, the easy reason, is that one of them is much larger because there are many more synapses between the two cells. So to determine how many synapses there are or how many release sites, what is the quantal size, what is the response upon the release of a single vesicle, and what is the probability with which the vesicles get released, then you have to do this so-called quantal analysis, and I don't have time to go into details, but we did it on lots of cells, and we ended up with a PV of 0.4. Now, again, it's very remarkable what Erwin Neher showed, that he also believes that in the calyx of hell, the mean release probability is 0.4. This is not exactly the same, and I can come back to this issue later. And the number of release sites are 10, and the quantal size is about 32. So when we look at this other connection, okay, which is facilitating a very small response amplitude, basically what we can see is that the probability of release in 2 millimolar calcium concentration is about 10-fold lower. There is a 2-fold difference in the N, and the quantal size is roughly the same. So basically, the two different synapses, there are slightly different number of release sites, but the fundamental difference is a presynaptic release probability difference. So the presynaptic axon of a pyramidal cell, whenever it releases transmitter, it releases in a way that it looks at who is the postsynaptic target, and the postsynaptic target somehow signals back to change the probability with which release occurs at this synapse. So this phenomenon is so fundamental, and I can talk a lot about, or I could talk a lot about it, how it influences cortical network computation as well. But I don't have time today. So the question is that what could be the most obvious reason for this difference? Now, since the discovery of Sir Bernard Katz about 50, 60 years ago, that neurotransmitter release is dependent on the calcium concentration, you can come up easily, the easiest explanation is that in one of these active zones, there are no calcium channels, and in the other one, there are lots of calcium channels. Or the second is that although there are calcium channels, for some reason, those calcium channels don't conduct. Okay, their opening probability or the channel conductance is low. Or alternatively, and that's again reflected by Professor Neher, if the calcium channels and the vesicles are physically far away from each other, calcium channels conduct calcium, the calcium concentration goes up, but wherever the release sensor is on the vesicle is so far away that it cannot really see that high calcium. So with another word, there is a large coupling distance between the voltage-gated calcium channels and the calcium sensor on the vesicle. So today, in the next, I don't know how many minutes I have, I would like to show you some data where we addressed all of these questions one by one. So what about the calcium channel? So we determined that with a method called immunolabeling on freeze fracture replica immunolabeling on brain tissue, and that was done by Andrea Lorenz, who is sitting here in the first row. And basically, we look at these active zones, and these black dots label the CAV 2.1 calcium channel in one type of synapse, in the other type of synapse. And basically, when we compare that in a previous publication a few years ago, we found only a very slight difference in the calcium channel number. The CAV 2.2 or N-type calcium channel number was identical. Now we repeated that. Andrea did that very recently in adult mice, and then again, she found a 14% difference in the calcium channel number. So there is this tremendous large difference in the release probability, but the total calcium channel number is seemingly the same. So I think we can rule out. What about calcium entry? Now, what we did, again, in a publication a few years ago, we patched onto these hippocampal pyramidal cells, and through the pipette, we inserted calcium-sensitive fluorescent dyes. And in the boutons, we scanned the action potential evoked calcium transients. And after the experiment, we fixed the slices, developed the biocytin in the cell, and we looked at the postsynaptic targets, whether they were parvalbumin positive interneurons or in this mGlu1 alpha positive interneuron, which is just a molecular marker for the OLM cells, for the somatostatin positive cells. And once we looked at it, actually we found a small difference in the calcium influx into the bouton. Okay? We repeated this again in adult mice in the CA1 region, and we found a 38% larger calcium influx into these parvalbumin positive. So the release probability is higher onto the parvalbumin positive cells, but the calcium concentration is also larger. So have we found the reason? Now, we try to address that using pharmacology. So basically what we did, the calcium concentration in the bouton is such in these mGlu1 alpha positive targeting cells, and this is larger in the parvalbumin targeting. But we used the drug, it's a potassium channel blocker 4AP, to increase the calcium influx, and we looked at what happens to the postsynaptic response. And this is the summary. So whenever we look at these guys, their concentration is 0.16, and the response is very small, the postsynaptic response. In these parvalbumin positive dendrite targeting boutons, the calcium concentration is 40% larger, and the response is 150 picoamp. So when we increased the calcium entry into these boutons using this drug, then we increased this concentration larger than this, okay, and we looked at the effect on the postsynaptic response. And on the postsynaptic response, we found an increase, but it's only a double the response, okay? So by fluxing more calcium into the bouton, we could increase the postsynaptic response, but we increased the postsynaptic response twofold instead of tenfold. So our data suggest that there is a smaller calcium entry, but there is a small difference only which is responsible for this PV. So it has to be something else. And then came this last part of the study where we try to estimate the distance between these calcium channels and the vesicles. And I'm talking about now distances. In this whole active zone is about a few hundred nanometers where the calcium channels are localized and also the vesicles, and these distances are actually nanometer distances, tens of nanometer distances. And again, I have to go back to a seminal paper published again more than 20 years ago, and to my knowledge, that was the last paper of Bert Sakmann and Erwin Neher together, but I'm probably wrong. And with Neil Bordashev and Rozov, they looked at this connection in the neocortex, again, a pyramidal cell innervating these bitufted neurons and these multipolar neurons. And it's beautifully shown that, you know, it facilitates and this depresses. And to start with, they looked at whether different calcium channels were responsible or not, and they found that drugs that block different types of calcium channels have very similar effect, so that wasn't. But when they did something, they used a calcium buffer. It's called EGTA, and the mechanism is not really important today. But basically, when they used 1 millimolar EGTA in this facilitating synapse, the facilitation disappeared. This is the important point here. And the same concentration of the drug, whenever they looked at this depressing synapse, it didn't touch the short-term plasticity. And then, in their paper, I would like to cite, they said that moreover, assuming that calcium sensors in the two types of terminals have the same high affinity, but the sensors are located more distally from the calcium channels, the pyramidal cell to bite off its cell at contacts, they could explain the rest of their data, including the effect of buffers or EGTA. And this is the schematic, what they drew. So there are vesicles and calcium channels, and these synapses, they are far away, and they are very close to each other, okay? And that is, I really like that. And then, of course, this is very, very obvious to test that experimentally, but experimentally it's not very easy. But before I show our data, I would like to point out a paper we published 2 years ago, where we looked at in the cerebellum 2 different synapses. There are these parallel fiber synapses where the release is low, it's a weak synapse, and the GABAergic interneuron synapse that the release probability is much higher. And then obviously we thought that, if you just look at the calcium transient in these boutons, the strong synapse will have large calcium transient and the weak synapse will have small calcium transient. And to our surprise, it was totally different. The calcium transient was 5 times larger in the weak synapse than in the strong synapse. And then, again, with Andrea Lorenz, and then we did lots of experiments, and mainly high-resolution localization experiment, and we came out with this model that although in this weak synapse there are lots of calcium channels, these are the red dots in the schematic, but they are pushed away from the vesicles, about 50 nanometer, whereas in the stronger synapse, much fewer calcium channels, but they are strategically located next to the vesicles. 2 minutes? Yeah. And basically they are close to each other. So this is a nice example that you cannot really predict the function just by looking at the calcium channel numbers. You have to know where they are. And I don't have really time to finish this, but Andrea did calcium channel localization and the protein, the MUNC13 protein, which is responsible for the function. And basically, these are the synapses. These are the clusters of MUNC13 molecules. That's where the vesicle sits. And basically, Andrea found that the calcium channels are actually outside of this region. And then she modeled this with the vesicles and the calcium channels, and then she excluded an area, but we call it exclusion zone, larger and larger and larger, and looked at, with modeling, that what is the distance which is responsible or could be explain our data, and basically what we found, and if the calcium channels are pushed away around 40 to 50 nanometer from the vesicles, then the data can be explained. But interestingly, that was exactly the same with the 2 types of synapses. So our data suggests that the nanotopology and the coupling distance between the calcium channels and the synaptic vesicles are not different. So basically, what is the reason for the difference? We don't really know, but we have some experiments which I don't have time to show today, where we also came to the conclusion exactly what Professor Neher said, that the MUNC13 molecule and the regulation of MUNC13 molecule is probably the key, and the vesicles in these 2 fundamentally differently working synapses are differentially regulated through the priming through MUNC13 molecules. Thank you very much for your attention.
Thank you very much for this excellent talk. Please stay on stage for